Hello, my name is Alex Pellegrini, and this is my Trypsin lab. So, what we were testing is a range of temperatures to see if there was an optimum temperature for trypsin reactions to occur. So we tested from a range of 47 to 77 degrees Celsius, going up 5 degrees Celsius per test, which gave us a total of 7 different results. These are our results right here. We did triplicate experiments, so 3 different tests for each temperature setting, and as well as a negative control, which helped us normalize the average, and we had pretty low standard deviations across the board. So the question that we were testing was, how will change in temperature affect trypsin activity? And we thought that the optimum temperature would be between 47 and 77 degrees Celsius. We were, in fact, right, as the optimum temperature was approximately 52 degrees Celsius. So, trypsin is an enzyme in the body that catalyzes the hydrolysis of peptide bonds and breaks down proteins into smaller peptides. Trypsin mainly cleaves to peptide chains, mostly on the carboxyl side of amino acids. This is also trypsin's role in live organisms. It is produced in the pancreas when stimulated and secreted by pancreatic duct into the intestine, specifically duodenum. The main purpose, as explained, is to break down proteins so they can be more easily absorbed into the body. So trypsin is found in animals' small intestine, like there. Humans' trypsin's optimal functioning temperature is around 37 degrees Celsius, but also depending on the organism's habitat located, trypsin temperature can vary. To determine the optimal operating temperature of trypsin, we conducted different sets of experiments multiple times at increments of temperatures to see which optimal temperature trypsin could thrive in. My personal abstract is the role of trypsin is to hydrolyze proteins. It is an enzyme commonly found in the small intestine of numerous vertebrates. Our results, okay, so other than the range of 7 temperatures, we also tested trypsin at with a buffer of 0.15 molar sodium chloride, 20 small m, big M trypsin hydrochloride at a pH of 8. All our results did not vary as much as we expected. However, our results did support our hypothesis, as I said earlier, at approximately 52 degrees. The t-test results for the optimum temperature of 52 degrees show that there is no significant difference between the results ranging from 47 to 62. No significant difference, as the p-values indicate. And from 52 to 77, there is a significant difference. So this is the key t-test. And these are t-tests for every single temperature that we tested. Although this is the one for 52 degrees, which we found to be the optimal temperature. So, in order to do this experiment, we used a micropipette that was 20 to 200 microliters to transfer 50 microliters of trypsin to a clean centrifuge tube. Then used the 100 to 1,000 microliter micropipette to transfer 500 microliters of azocasein to the same centrifuge tube containing trypsin. We next used the micropipette tip to get a new one to obtain 450 microliters of reaction buffer and transfer it to the centrifuge tube. We repeated this 7 times for each temperature value, and then we triplicate, and then because we performed triplicate experiment, we performed it 3 times for each temperature. So then we placed all the temperatures away and incubated them for 20 minutes, and we also placed them in the centrifuge to isolate the pellet and extract the supernatant. We use 100 to 1,000 microliter pipette to add 500 microliters of TCA. We wore gloves because TCA is dangerous. Microcentrifuge tubes, we at max speed for 200 minutes, so that was at 14,000 RPMs per minute. We transferred the supernatant to a clean cuvette, and then we blanked the spectrophotometer to 440 nanometers using a cuvette filled with distilled water, and the spectrophotometer is it measures UV light rays. So then we read the absorbances and we copied them down, which is all this right here. So this right here is our graph. On the left, it shows the normalized average, which is basically the triplicate experiment for each temperature with the negative control value added in. So as you can see, this is the experimental average, which is shown in this graph right here. And this is the normalized average right here, which is shown in this graph right here. And so basically, this big gray bar represents the value, and this little funky black bar represents the error bar. So it represents basically what we see in the t-test results and the p-values, which is if there was a significant difference or not. So for example, as I said earlier, we could not determine that 52 degrees Celsius had a significant difference from 47 to 62 degrees, so we cannot say that our hypothesis was 100% correct, although it did prove to be the highest, it did prove to peak. However, the error bars overlap, so it renders our results we don't know if they were right or not, basically. So let's discuss. The results do support our hypothesis. However, they do support our hypothesis that there will be an optimum temperature between 47 and 77 degrees Celsius for trypsin reactions to occur. However, the optimum temperature for trypsin reactions to occur is approximately 52 degrees Celsius. We cannot state that the data, as I said earlier, that from 52 degrees Celsius is significantly different from the data for 47 degrees Celsius, 57 degrees Celsius, or 62 degrees Celsius, because the error bars do overlap, as I showed right here. Right here. The p-values, when comparing 52 degrees Celsius to 47 and 57 and 62 degrees Celsius, are all greater than 0.05. The absorbance between 47 and 77 degrees Celsius differs by approximately negative 28.5%. The absorbance between 52 degrees Celsius and 57 degrees Celsius, which is the closest absorbance level to that of 52 degrees Celsius, is negative. It differs by approximately negative 11.2%. The variability among trials was low, and the standard deviations, shown right here, only ranged from approximately 0.6 to 15%. The other group's data also, the other group who did the temperatures, also showed 52 degrees Celsius was the optimum temperature for trypsin reactions to occur. Their error bars also overlapped at 52 and 57 degrees Celsius, Therefore, trypsin's optimal activity rate is in between 52 and 57 degrees Celsius, which does support our hypothesis yet again. The ideal condition to grow bacteria, the ideal condition, X, for trypsin reactions to occur is at about 52 degrees Celsius. These are our references cited. And I just want to go back and explain that throughout all this, as we added the TCA, that was so that we could stop the reaction. We centrifuged it to create supernatant and the pellet, as I explained. Thank you very much.